All right, thank you. I think we can get started. So it's a pleasure for me to welcome you all to this workshop, which is part of the Precision RNA Medicine Core's workshops on technologies that are useful for RNA research. Today, we will have presentations from our core facility, in particular the detection unit, and then one of our partners, Mirexis, will talk about a new technology that they're offering through our core Costeria-seq, which we think will be a very useful technology for RNA research. So just by way of introduction, my name is Frank Slack. I'm the co-director of the Precision RNA Medicine Core here at BIDMC. Our first presenter today is going to be Dr. Jan Heng. Dr. Heng is an assistant professor of pathology at BIDMC and Harvard Medical School, and an affiliate, so I should mention that BIDMC is an affiliate of Harvard Medical School. She conducts translational breast cancer research, and in particular, she's developing clinically relevant modalities for breast cancer patients. She's a native of Singapore, where she completed undergraduate and PhD studies. She completed undergraduate and PhD studies in Australia, and then trained at the University of Toronto, Canada, before joining BIDMC as a principal investigator. She's also part of the Cancer Genome Atlas Breast Cancer Working Group and is a long-time collaborator of the Nurses Health Study for Molecular Pathology Epidemiological Research, and leads the BIDMC Transgender Breast Cancer Working Group. So it's a pleasure for me to introduce you to Jan, and Jan will start the session.
Thank you, Frank. Just confirm, can everybody see my slides?
Yes.
Thank you. So thank you, Frank. So as I mentioned, I am the principal investigator. I'm a principal investigator at BIDMC. We are an affiliate hospital with Harvard Medical School. I particularly conduct translational breast cancer research in cisgender and transgender populations. I'm also part of the Precision RNA Medicine Core housed at BIDMC. Our core has 4 units: detection, spatial, bioinformatics, and delivery. And I head the detection unit, highlighted in yellow. So our detection unit is Mirexis, which is the company that is partnering with us for this workshop today. We are the USA flagship service core, which means that if you're in the USA and you would like to screen your samples for microRNAs using their assays, you will be referred to us. Mirexis offers 3 microRNA screening panels. I'm introducing these panels so that if you could get these kits and do them in-house yourself, but if you don't want to, we can do it for you. We'll order any of these kits and we'll run them for you in our core. The 3 kits that they offer are the biofluid microRNA panel that screens for 176 microRNAs. Cancer panel screens for 352 microRNAs commonly implicated in cancer, and Panomere, which is their most extensive panel, screens for 376. Mirexis can manufacture custom panels if needed, or also offer any microRNAs as individual assays. And Mirexis has ready-made validated assays for 2,600 human microRNAs. So for my research, I use the cancer microRNA panel, and this is the panel I use for the research I'm presenting. And the aim of my presentation today is to take you along my project from conception to data analysis so that you get a sense of how this technology can contribute to your research. As I mentioned, BIDMC, we are one of the many teaching hospitals of Harvard. And at BIDMC, we have an integrated multidisciplinary approach where scientists and clinicians collaborate in many research areas. And thanks to Frank and our hospital researchers were the first to have access to Mirexis microRNA screening panels in the USA. So Dr. Gerber-Wolf, and she's shown here, and I leveraged that privilege to explore microRNAs as a clinical tool for breast cancer. Dr. Wolf is the scientific director of Phase 1 clinical trial program at BIDMC, and she's also my breast cancer mentor. So our study aims to investigate whether plasma microRNAs can potentially predict response to breast cancer treatment. So this trial is known as the INFORM trial, and it was published in the Journal of Clinical Oncology in 2020. Now, this trial has the perfect study design and defined clinical outcomes for us to explore microRNAs as a clinical tool for breast cancer, and whether plasma microRNAs can predict response to breast cancer treatment. This trial is led by our own Dr. Nadine Tang, and we're grateful to her for giving us permission to evaluate her patient's plasma. In this trial, we had 118 patients enrolled, and in particular, they had germline BRCA1 or BRCA2 mutations, and most of them have the estrogen receptor negative subtype of breast cancer, or some they could also be classified as triple negative breast cancer. So I'm just going over quickly about the trial, briefly, obviously. So this trial, this INFORM trial, was conducted across 13 hospitals in the USA. When patients are recruited, we obtain their EDTA blood sample and tumor core biopsy before starting treatment. And then the patients were then randomized by their estrogen receptor status and hospital site. And they were either randomized to receive mono platinum therapy, which is neoadjuvant cisplatin, or they were to receive standard chemotherapy of doxorubicin and cyclophosphoramide, or we abbreviate it as AC. And after 4 cycles of protocol-assigned chemotherapy, the patients would undergo breast surgery. And so after undergoing surgery, the tumors were then assessed for residual cancer. The primary outcome for this INFORM trial is known as pathological complete response, or PCR, which is defined as the absence of residual invasive disease. And PCR is then classified as to, did they achieve PCR, which means no more tumor is detected in the patient, or did not achieve PCR, which means the patient still has residual tumor. And so we leverage on this plasma collected before they went on before they were randomized to the treatment arms. And then our hypothesis is whether plasma microRNAs are associated with achieving complete pathological response. So of the 118 patients that were enrolled into the trial, 99 patients completed 4 cycles of treatment and had pre-treatment blood samples. So we got an aliquot of these plasma. We aliquot 250 microliters of plasma, and we send the plasma to our BIDMC Precision Medicine Core detection unit. And then the core it's a full service core, right? So the core performs RNA extraction and runs the cancer panel, which screens for 352 microRNAs. It is 1 patient per plate. And for 99 samples, we screen the samples over 13 days. So this is just a quick, simple overview of our pre-processing workflow to clean up the data before statistical analysis. So if you use this technology and you send the samples to us, when we finish running the assay, we'll send you back the raw microRNA data, which is CT values. So then on your end, you would then perform QC checks. You would check the RNA spike-ins in specific control wells. Then you would exclude data where CT values are less than 8 or 9 or greater than 33, like you would when you do PCR assays. You would perform the first normalization of the data across per plate. So you normalize within the patient. In step 5, you exclude lowly expressed microRNAs. Okay, and then in step 6, you would then identify housekeeping genes specific to your samples and then normalize your data again using the expression of these housekeeping genes. And for me, because I had so many samples, and our samples were screened over 13 days, I observed batch effects. So I used a computational method to correct for these batch effects, and you can choose whatever method you want to correct for batch effects. And then at the end of this pre-processing workflow, we had 97 samples left, 53 that patients who received cisplatin, 44 received chemotherapy, and 251 microRNAs for analysis. Now, you can use any statistical method to analyze your data. For me, I use binary logistic regression because my outcome was, did they achieve PCR or not? And then I also, after evaluating the data, I decided to stratify my patients by treatment. So I analyzed within cisplatin patients and then within chemotherapy patients. So I analyzed whether each of the 251 microRNA was associated with PCR. And using the false discovery rate of 0.05, I didn't see any microRNA associated with complete pathological response. And that's not surprising because we only have 50 samples in each treatment arm, but we have 251 hypotheses. So more hypotheses than samples. So it is not surprising we didn't identify any microRNAs at this stringent FDR less than 0.05 cutoff. This table here shows you the number of microRNAs, so the number of microRNAs that pass various statistical cutoffs from P less than 0.05 to FDR 0.25. Now, if you look at this table, something is very striking, right? So our clinicians themselves were very stunned to see that there was no microRNA signal associated with complete pathological response among patients who received standard chemotherapy, but there was some rather strong response or signal among patients that received cisplatin. So using the next most reasonable cutoff, so we use P less than 0.01 and FDR less than 0.25, we found 8 microRNAs, and they were all upregulated in patients who achieved pathological response, and specifically in the arm of patients who received cisplatin. And then the results for these 8 microRNAs remain significant even after controlling for clinical variables such as the age, how old the samples are, triple negative status, the grade, stage, and the how much tumor infiltrating lymphocytes were present in the tumor. And now, due to invention disclosure, I do apologize that I am unable to reveal the identities of these microRNAs. And so we think that these 8 microRNAs in the plasma may reflect tumor characteristics or unique tumor characteristics or the patient's immune response that actually confers cisplatin sensitivity. And now, cisplatin is not a common choice of drug to treat breast cancer, but it's useful. It's potentially useful to know that this patient's tumor could be sensitive to cisplatin, and it could, it might be something for the clinicians to consider to give cisplatin in a neoadjuvant setting, which is before surgery, or when a breast cancer patient has exhausted all their options when they have metastatic cancer. So in the next steps, what we're doing now is we're performing in vitro functional validation of the identified microRNAs. And so platinum-based therapy is actually more commonly used for lung and ovarian cancer. And so a step in the future would be explore if any of these 8 microRNAs can predict or is associated with response to therapy in lung and ovarian cancer patients. And so before I end my talk, I'm going to do a quick plug again that our detection unit, which is what I head, is a full service core to help you screen for microRNAs in your biological samples. These are the biological samples that we have established protocols for at the moment. And so we will work with you on sample collection, submission. We will perform RNA extraction and the screening, microRNA screening of your samples using any of Mirex's panels. And then when you get your data, Mirex's will at least provide you with some basic pre-processing support before you do your statistical analysis for your biological question. And so with this, I end my presentation by acknowledging my amazing colleagues, Dr. Wolf, Shizong, Lin, Dr. Nadine Tang, Felipe, these are doctors too, they are postdocs, and the Precision Medicine Core, Frank, and our staff scientists, the Informed Trial Committee members, and group at Mirexis who will help us with pre-processing steps and our funding resources. Thank you, and I will stop here.
Thank you very much, Jan. Appreciate that. So now we have a chance for people to ask questions. If you would like to ask a question, please could you type your question into the Q&A box, or if you raise your hand, then Grace could manually unmute you and you could ask your question. But perhaps for now, let's see if there are questions in the Q&A box. Okay, well, maybe while people are typing, I could ask you a question. Maybe you could provide some information about the kinds of analytical tools that are available for users for the data pre-processing. I know you mentioned that Mirexis does some of that, but what's available to users?
When you receive your raw data, Mirexis has a nice Excel sheet, like a worksheet for you to plug your data in and explore it. So that's one way to go. And if you are code savvy, they can also provide you with an R package. They actually are providing, they are making an R package. They have an R package to guide you through cleaning up the data until it reaches a statistical, ready for statistical analysis. So an Excel sheet or an R package. It's pretty useful.
Okay, fantastic. We have some questions in the Q&A from an anonymous attendee. What is the turnaround time for the supplied samples from when you supply the samples to when you receive the data? And I guess you know, for a typical sample set of about 50, what do you think the turnaround time would be?
Right. So for 50 samples, I think the turnaround time would be about 3 to 4 weeks, because it takes the staff to do all the RNA extraction and then running the plates and collecting data. So 3 to 4 weeks for about 50 clinical samples.
Okay. We have a question from Purya Nadiri. Can you please explain what determined Mirexis batches that you needed to correct? Was it based on plasma collection or based on Mirexis plate preparation?
Thank you, Puri. It is based on the plate preparation. I think we found, so using the spiking RNA, we observed batches between the days. Yeah, so it fluctuates by which day the samples which batches of samples run per day. And so I adjusted according to day, so 13 days.
Okay, so it's not the date of the plasma collection or the RNA extraction, it's the date the assay was run on the plate.
Yes.
Okay. We have a question from Matthew Waldor. Are there pipelines for murine microRNAs, and how about non-targeted screens? I'm not sure about that last question.
Yeah, so at the moment, Mirexis does not have assays for murine microRNAs, but Frank, aren't they highly conserved?
Yeah, many of them are highly conserved. So we've run murine samples through the human plates.
That's right. I did that too. I ran 4 mouse breast cancers. Yeah. So if your microRNA of interest is highly conserved between mouse and humans, it is possible. And then we'll convert. And then we'll convert whatever that was. We will convert and give you the results for the mouse equivalent microRNA, which we have done before. Yeah. Okay. Yes. Thank you, Matthew.
Matthew, I'd appreciate if you could just clarify in a separate question what you meant by non-targeted screens. We'll come back to that. A question from Dara Sachdev. How much RNA is required per patient to perform the assay per plate?
So, Madison, are you on the call? Let me unmute her, yes.
Madison, you should be able to speak. I just unmuted you. Okay, yeah. So I don't have it off of the top of my head, but we do have a form I could send out that like lists all of the requirements for all of the samples. If you already have the RNA, obviously it takes out a step, which makes it a little bit easier for us, but we can also just do it straight from the sample. I believe it's like 50 nanograms, but like I said, don't hold me to that.
Thank you,
Okay,
Madison.
thank you. We have a question from Louis Christodoulou. Sorry, I'm messing that up. What accreditation does your lab have? For example, is it CLIA or CAP or ISO? For example, does it have the potential to run clinical trial samples? And are these panels FDA cleared or approved?
So I would direct this to Craig. Craig, would you like to answer this?
What?
Yeah. Hi. I'm sorry. Go ahead, Frank.
Oh, no, I was going to say, I can tell you that our core facility currently right now is not CLIA approved, but we'll turn it over to Craig to answer the question about whether the panels are FDA approved. Go ahead, Craig.
Yeah. So we have. Well, the panels are for research. The ones that Jan recommended. So those are all research panels. But as a company, we do have a CLIA lab, but we have several labs in Singapore, and we do run clinical samples out of the labs in Singapore that have been approved through the agencies in Singapore, and one panel is the GastroClear for gastric cancer. So we do have the capabilities in Asia out of Singapore lab to run clinical samples.
Yeah, so just to reiterate, the company itself has a approved clinical test called GastroClear to screen for gastric cancer in Asia, but at the moment none in the US. Thank you, Louis. And we have a last one.
Great. Another question from Celia Brochin. She says, hi, thank you for the presentation. Were you able to have plasma before the chemotherapy to analyze the levels of microRNAs you identified before the treatment to predict the response?
Yes, yes. So the samples that we ran, all our 99 samples that we ran were all before chemotherapy. So they were pre-treatment. So we collected the blood first. They got randomized to receive cisplatin or AC, and then we knew the outcome, and then we correlate pre-treatment levels to outcomes. Thank you.
Great. So actually, Matthew did clarify what non-targeted means. He's really just wants to know whether there are ways to basically select the microRNAs that you'd like to be screening for to make a custom panel. And I believe that is possible, but maybe we can also ask Craig to clarify on that, whether Miraxis would make a custom panel for an individual client.
Yes, thanks everyone. Yes, no, we do custom panels. As Jan mentioned, we have our set panels, the 3 set panels that we have, and then we'll customize panels based upon the need of the client. But yeah, we can customize panels for clients.
All right, so yeah, you can work through us, and we will put you in touch with Miraxis to get those panels created.
Yeah,
great. And then I think one more question from Ming Tao. How do you evaluate the sample's quality before you proceed further, especially for patient specimens? And she's also asking, how long can the specimens be stored at -80 before the process?
So I'm assuming how long the samples he's referring to clinical samples, like how long did we store the clinical samples before batch processing? Mm-hmm. These are clinical samples. They were collected between 2012 and 2017, and then RNA extraction maybe a year later. So the samples were stored for about a year. They're clinical samples. We really can't control this. I mean, the best way forward is to incorporate this before you start the trial to make sure you control everything. But to answer that question, is it will store at least for a year in -80. And how do you evaluate patient quality? So, Madison, what kind of quality of the RNA do we check before we put them into the assay?
Yeah, so we normally do a nano drop just to make sure that there's not any RNA degradation or anything like that. As far as like patient samples go, we want to make sure obviously like the serum samples are the best quality serum we can get. We can run the samples with lower quality serum, but we have seen that the worse the serum quality is, the worse results we get, and it's so just trying to make sure that those samples are, obviously the best quality samples are going to be the get best quality results.
So when we run clinical samples for clients, we run the RNA first, and then we check in with them to confirm whether they want to proceed or not, right? Based on the RNA
Yes.
quality.
Mhm.
Yes. Thank you,
All right,
Madison.
and Madison, do you want to clarify how much RNA is needed per sample?
Yeah, so I was able to pull up our sheet. So yeah, generally we say like the 50 nanograms per microliter with at least 20 microliters, or just in general, the 3 micrograms. So somewhere around there. Obviously more is better, but that's like our minimum.
All right, thank you very much. I think we're gonna close this part of the workshop, and I'm gonna turn it over to Jan, who'll introduce the next speaker. Thanks, Jan.
Thank you. Thank you, Frank. And so it's now my pleasure to moderate the next talk. And as a reminder, you're welcome to actually type your questions in the chat box anytime. And we apologize in advance if we are unable to address your question due to time constraints, if it happens. And also, feel free to email me, Dr. Kumar, who's next, Mirexis USA, so Craig Morley, if you have any follow-up questions. So I'm so happy to introduce Dr. Vikran Kumar. Dr. Kumar, would you like to put your slides up, like prepare your slides?
Sure.
Thank you. So Dr. Kumar is the head of bioinformatics and the associate director of precision genomics at Mirexis. He has 20 years of experience in the field of human genetics. After completing his PhD from the Indian Statistical Institute, he joined the Genome Institute of Singapore to study the association between host genetic variation and susceptibility to infectious diseases. After GIS, he joined the Duke and National University of Singapore Medical School and actually started the single-cell and spatial transcriptomic program in gastric cancer. And he joined Mirexis a year ago and leads the genomic bioinformatics unit. He leads the development of pipelines for multi-omics data integration and data analysis, including analyzing spatial transcriptomic data generated using this new stereostic technology, which he's going to introduce today. Thank you, and Dr. Kumar, please.
Thank you, Dr. Han, for your kind introduction. Let me start with my video. Can you guys see my video?
Yes.
Good morning, all. Today I shall talk about StereoSeq. It is an acronym for Spatial Enhanced Resolution Omic Sequencing. Using this technology, we can profile whole transcriptomic at single cell level in an unbiased way. In this talk, I shall first briefly introduce you to spatial technology, workflow, followed by its advantages, and demonstrate some of its applications. Most of us are now well versed with single cell technology and its advantage over bulk RNA sequencing. It allows annotation of each cell. However, the spatial context of the cell is lost. Using spatial technology, we are able to preserve the spatial context, which provides biology a meaningful context. For instance, we may want to compare the macrophages expression in core of the tumor versus macrophages expression on the margin of the tissue. This information is not possible to have from single cell. Spatial technology in the form of single probe began in 1982 and has continuously evolved over a period. From single probe, it has moved to multiple probe, and now whole transcriptomic profiling can be done. With StereoSeq, whole transcriptomic profile can be done at single cell level at a very unprecedented resolution. In StereoSeq, we do not need special equipment except a sequencer. A patented chip with millions of spots has DNA nanoballs. These are the DNA nanoballs, and which is made up of poly T, UMI, and CID. Poly T is to capture poly A RNAs, and CID has X and Y coordinates of each location. Once on this particular patented chip, a tissue is fixed, permealized, RNA is captured, library is made, and then the data analysis happens. Now let me discuss the workflow for StereoSeq, which is very simple. There are two workflows, one is F1 and another is S1. F1 workflow is to figure out the optimal permealization time. We cannot have a very short permealization time, otherwise we may not be able to pick up all the RNA, and we cannot have a very long permealization time, otherwise most of the RNA will leak out from the cell. So in this F1 workflow, there's in the chip, we fix the tissue, and then at different time points, we look at the permealization time testing. And for each time point, we do a reverse transcription, cDNA is prepared, tissue is removed, and then we do imaging of that cDNA. And depending on the content of the cDNA, we can figure out whether the permealization timing is correct or not. Once the permealization time is fixed or it is optimized, we then put the tissue on the S1 workflow, which is a similar workflow where you put the tissue on the chip, and then the tissue staining happens and the imaging takes place after staining. In this tissue staining, primarily we stain the nucleus, and then tissue permeation happens based on the F1 workflow, and then cDNA is prepared, collected, library is prepared and amplified for sequencing, and then sequencing happens. Let's look at the data analysis now. It requires 4 different kind of files. Number one, we require FASTQ file, which contains all the sequencing data generated by the sequencer. We require a mask file, which contains all the coordinates of the spots, which are those X and Y coordinates of the spots. We require a reference genome, depending on tissue coming from which kind of organism. And then we require the image where we did the nuclear staining. Based on this 4 input file, we can run the pipeline. We map the sequences, we merge and generate a gene count matrix, and then the tissue is registered after that. And we also do a tissue cut if there are multiple tissue fixed on the same chip. You can do multiple tissue on the same chip because your tissue size, your chip size is quite big. And once this all happens, then you are good to go for tertiary analysis. There are programs available for StereoSeq, particularly to do some of the analysis using StereoPie. There is another program called SAW, and then you can use using these programs, you can even visualize the data at single cell level. You can also use third-party programs like Spatio to do cell segmentation. And once you are able to assign all the RNAs to a cell, then you can do all kind of analysis using even Surat or any third-party programs like InfraCNV, keeping in mind its spatial location. Let me elucidate some of the advantages of this StereoSeq technology. StereoSeq technology allows a very high level subcellular resolution, which in turns allowed you to do a very good single cell segmentation. Now, if you see this graph here, the white color is nucleus and the green color is the RNA captured in that particular cell. And using nucleus and technology and the algorithms like Spatio, one can do a single cell segmentation. On the right side, each of the cell is colored by different color based on the annotation of that cell. Important feature of stereosec is a large field of view, which allows you to capture much more region of a tissue. It starts from 0.65 centimeter into 0.6 centimeter. All the way you can go to 13.2 centimeter into 13.2, where you can even fix half of a human brain. Already a paper is out where 3 centimeter into 5 centimeter chip is used for macaque brain. We in-house regularly use 1 centimeter by 1 centimeter, which is very good. And even for that, we do a lot of sequencing, almost like 1.5 billion reads. And if we do bigger ones, there would be insane amount of sequencing will be required. Because of its high resolution and field of view, you can compare stereosec resolution versus resolution coming from another technology. I would not name this technology, but perhaps you might be able to figure it out. And you can very clearly see the level of resolution coming from stereosec compared to other kind of technology. Let me demonstrate some of the application where stereosec was used. Stereosec came very recently, just 2 years back, and last year there were papers in high impact factor journals like in Cell and Science and other journals like Developmental Science, which demonstrated different and broad application of stereosec technology. In this particular paper, the authors used stereosec to reconstruct spatial and temporal transcriptomic dynamics, which happens during mouse organogenesis. And they were able to show that at a very high level cellular resolution. They picked up 8 stages with 53 sections from embryonic 9.5 days to 16.5 days. They could show cell type heterogeneity, and they could also identify previously unidentified cell types or cell states. They were also able to decipher developmental disease susceptibility. For example, using mouse embryo development, the authors were able to show 2 particular genetic diseases which happens in human. One is Robinow syndrome, where a mutation in Wnt5a gene causes dysmorphic face and limb. And using mouse embryo development, they were able to show the expression of this gene only in the limb and maxilla. And as I said, this is a problem of dysmorphic face and limb. Similarly, in brain-lung thyroid syndrome, which happens because of a mutation in NKX21 gene, the authors were able to show its expression in lung and hypothalamus, and it's known that in this particular syndrome, a person suffers because of respiratory and thyroid problem. Our own in-house data generated on B-cell lymphoma using stereosec technology. 2 B-cell lymphoma tissues were profiled in our lab. Left side image are the H&E stain ones, and the right side image shows tumor cells with percentage of genetic alterations estimated using stereosec poly-RNA dataset. We used InfraCNV algorithm to estimate CNVs. Cells with high proportion of genetic alterations are indicated by sets of red. The first tissue with pink mark are supposed to have tumor cells. Indeed, the right side image shows cells with red color in the same region, showing a good correspondence between the image generated by the stereosec dataset and the H&E stain image. The other region is devoid of such cell, and H&E stain suggests these regions also as non-tumor area. This is another example of using stereosec to profile cervical squamous cell carcinoma. And here they try to understand the immunological microenvironment, and then they subdivided the stromal area into cellular neighborhoods according to their lymphocyte infiltration status, which includes stroma with diffuse inflammation, and stroma with focal inflammation. They also try to characterize pro-tumorigenic myofibroblasts. Another example where the liver tumor was profiled, margin area, there were 8 samples from margin area was taken, And then the tumor tissue was also taken from the core. Lymph nodes were profiled, and then adjacent normal tissues were also profiled along with that. At this study, I would like to have your attention in this particular margin area. This is an H&E stain, and you can see on the left side of H&E, this is the tumor cell. On the right side is the non-tumor cells, and margin is very well delineated. Based on the stereosig data, you can see a very nice correspondence of the margin. It's very well recapitulate the margin. The left side is all full of tumor cell, and the right side is devoid of all kind of tumor cell. A similar thing you can see in the metastatic lymph node, where the top part is lymph cells or immune cells, and the bottom part is full of tumor cell, and that is also shown by the data generated through stereosig data. Same study, one could identify tertiary lymphocyte structure, which is not possible to do just using single cell transcriptomic analysis. Because we knew the spatial location of all the immune cells, the question was asked, where are the B cell, T, NK cells, and dendritic cells are co-aggregating? And those co-aggregated cells, those locations were termed as tertiary lymphocyte structure, and then its distribution across the tissues were figured out. Another application of stereosig was used to profile the anterior part of the brain of the axolotl. Here, the brain was induced with an injury, and using stereosig, they were able to capture the cell lineage transition, as you can see from here, during regeneration, and then they compare with the developmental process. Because of the high definition large field of stereosig technology, they were able to generate spatial transcriptomic data on single cell resolution from sections that cover both hemisphere of the anterior part of axolotl. In this particular example, this is the first time a 3D reconstruction of Drosophila late stage embryo and larva happened. In this study, every fifth section of the late stage embryo and larva was taken to construct a whole spatial transcriptome, and it was never done at such unprecedented level on Drosophila embryo and larva. This is an interesting study which could be possible using stereosig to look at cell-to-cell interaction. In this particular study, zebrafish model was used, where cell-to-cell interaction of Notch ligand receptor showed a very dynamic range over time and distance. If you look at this particular dot plot, and the y-axis is your time, and as time goes by, these red dots, the big red dots, indicates a very strong cell-to-cell communication of ligand receptor of Notch. Having demonstrated different application of stereosig, we in-house at our lab, we have done many different kind of tissue types and tumor type. For example, we did human cerebral organoids and mouse organoid too, where we could fix multiple organoids in the same chip 1 by 1 centimeter, since the organoids are small. Similarly, we did lymphoma tissues, as I showed in my previous slides, where we could put two tissues of lymphoma, B-cell lymphoma in the same chip. We also did a very interesting study on hair follicle and a study on liver tumor and normal. We are conducting right now a study on human placenta and hepatocellular carcinoma with colorectal cancer, nasopharyngeal carcinoma, heart arteries, and mouse brain. And thankfully, so far we have 100% success rate. None of our tissue have failed. Based on the data generated by our own lab, we wanted to quantify the impact of cDNA quality defined by the proportion of RNA with greater than 1000 base pair length. The left side scatter plot shows significant correlation between cDNA quality and the number of annotated reads. These annotated reads are uniquely mapped reads, and for each sample, we generated a total of 1.5 billion reads. On the right side is the scatter plot of cDNA quality and the total number of protein coding gene in the entire tissue. The cDNA quality has no correlation with the protein coding gene, suggesting that regardless of cDNA quality, total number of gene detected is not impacted. It also suggests, based on the cDNA profile and total annotated read, that the low quality cDNA can be salvaged. The data from the low quality cDNA can be salvaged by doing much more sequencing so that one can achieve at least 80% sequencing saturation. Over a period of time, we have also improvised our protocol. In the earlier version, if you look at the statistics, the uniquely mapped reads were close to 50% in all the samples, and there were a lot of multi-mapped reads, and there were some reads which were neither multi-mapped or uniquely reads. And the other thing, what we realize is that we still get a lot of ribosomes. Also, it is a poly A. We get a lot of mitochondrial RNA and a lot of housekeeping genes, which usually we discard while we analyze, unless until it is required for some specific purpose. In the version 2, improvised version, we in-house developed it. Now we are able to map almost 80% of the reads uniquely. The number of multiple mapped reads have gone down considerably, and so is the reads which are not human reads most likely. And in the process, also we have reduced our ribosomal RNA map reads, mitochondrial map reads, and housekeeping reads considerably from version 1. And that led us to increase our gene of interest from 86% to 94%. That's a huge jump in the number of genes detected. We have also come out with a protocol for multiplex immunofluorescence. This technology enables spatial visualization of multiple proteins with unbiased whole transcriptome information on the same tissue slide. Additional detected protein information can be integrated with gene expression data to in-depth evaluate valuable sample, and this has no bearing on the mRNA expression data at all. So this is one example of mouse thymus where with DAPI nuclear staining, we also did 3 antibodies, CD68, CD3, and CD45. And this helps in further analysis of the mRNA data. In the stereosync technology, permeabilization is very important. So we have tested permeabilization and successfully established for brain, thyroid cancer, nasopharyngeal cancer, lymph node, and all kind of different tissue types from human and as well as from mouse. And people also use mouse for xenograft, and therefore we were able to also do analysis from different tissue types of mouse. We were also able to do on different other kind of organisms like Drosophila, zebrafish, salamander, macaque, and some of them are used as model organisms as well to understand the biology. So this whole different kind of tissue types and organism allows us to do biology at a very unprecedented high level of resolution using stereosync. End of my presentation, and I would like to thank you for your kind attention, and I'm ready to take any question now.
Thank you, Dr. Kuma. That was a great introduction and overview of the stereosync capabilities. And so we're happy to answer questions now during the Q&A session. So we do have some questions in the chat Q&A box.
Dr. Kumar, your camera's off and you're still sharing, by the way.
Okay. Yep. Okay, thank you. Thanks for letting me know this. Okay. I'm not
Okay.
able to. Yeah. Can you hear me?
Yes, but your video is not on.
My video, I mean, I'm not able to start my video for some reason. It's saying host has stopped it. Yeah. Okay.
So so there's 4 questions in the chat box, and I'm just going to answer. There's more coming in. Awesome. I'm going to answer one very quickly. So I would just remind everybody that this session is recorded, and there is a link to all the presentation. So we will send you a follow-up email after this workshop, and there will be a link where you can access this workshop's presentation. And so this is to answer Hanwei's question, because he couldn't see your slides because it was pixelated. So you'll be able to review the video at the end of this. Thank you. And so, so the first question was Matthew. So, right. So Matthew was asking Dr. Kuma, how does StereoSeq technology compares to MERFISH?
StereoSeq is able to do whole transcriptomic analysis all the time for each cell. So MERFISH can do that as well, but scaling it up is a problem and it's expensive. And also when you're doing StereoSeq, we think that we are able to pick up big tissues. The field of view is much larger than you can do for other kind of technology available. MERFISH is also prone to index errors. If you have a lot of indexing error, that can impact your number of gene detected as well. Yeah, I would say so. Right, StereoSeq, we would like to make it as affordable as possible over a period of time so that people can use it more and more.
All right, so just on the same question, someone is Peya Aurora was also asked, so how does this then compare to NeoGenomics platform?
Oh, I'm not heard of new genomics platform, unfortunately.
Yeah, so
I'm just checking that out.
Right. I wonder if maybe that's, yeah. So Peya Aurora, if you could clarify what is NeoGenomics. Thank you. Okay, next question. How do you reduce the reads for ribosomal RNA genes, housekeeping genes, etc.?
Right. So this is our proprietary kind of protocol. I mean, I can just tell you that using a CRISPR-based method, we are able to do that. Unfortunately, I can't go into details of it, but we have a way where we apply CRISPR-based technology to remove ribosomal mitochondria and housekeeping. We don't remove all housekeeping. Some of them are interesting for cell cycle and other kind of stuff.
Okay, so Peya responded saying NeoGenomics platform. It says a single cell spatial platforms with Neo. Do you know what that means? No. I think Jason's going to answer that. So
yeah, I can answer that. I think she's referring to NeoGenomics, which is a company that uses NanoString exclusively. So maybe if you could just kind of just answer, based on which would be the GeoMax system.
That's what I thought. So she's referring. Peya is referring to GeoMX by NanoString.
Okay, so NanoString has both GeoMX and they have launched recently Cosmx. In GeoMX, you can use primarily 3 or 4 different stains, immunofluorescent stains, and then you have to pick up based on those stains where is a fibroblast and where is your, let's say, epithelial cell or endothelial cell, and then you mark those regions, use the help of a pathologist, and then sequence it. So they are not necessarily single cell because a large region of interest is picked up. It could be an aggregation of 20, 50, 70 cells, depending on what fluorescence you are picking up, and underlying that, they might have contamination of other kind of cells. So that's not truly a single cell in that sense. You have to do the deconvolution to show that they are all coming from one particular kind of cell type. Cosmic is another one, but they are not whole transcriptomic. You can do, I think, at the most 5,000 genes, and even in that 5,000 gene, not all can be done. I mean, you are able to get maybe 50 to 100, or that's the number of genes we are able to get per cell. I mean, I can't tell you exactly now what's happening on the Cosmics, but earlier it was tried, and the number of genes detected per cell was very, very low.
Thank you. There's just a lot of questions going on in the chat box, so I'm trying to keep up. Okay, so we have a question from Dara Sashdev. For the application of stereo-seq, genetic biomarkers should be well established. Can we also identify signature biomarker associated with the disease using stereo-seq data?
I think so. It will all depend on your study design, actually, and the kind of tissue you are going to use. That's possibility. I mean, this technology does not discriminate between different kind of diseases. It's up to you. How do you want to use that?
Thank you. We have a question from Ming Pao. Could you comment what is the general cost of this type of assay?
I would take help of my team members who are here who can answer this question.
And I thanks. No, it's a good question. I'll speak for North America pricing. So right now the list price per chip is $10,500 on the list price. And as Vikrant mentioned before, each chip is 1 centimeter by 1 centimeter. So figure about 90% of that real estate can be used. So depending on the sample size of how many samples you can get on the chip. And that is all the way through the slicing, preparation, imaging, and sequencing information coming back. That price. So again, list price is $10,500 per chip, and if you get more samples on it, price per sample goes down.
Continuing on that 1 centimeter by 1 centimeter chip, we have a question from Priya. Can you provide estimates of how many cells and how many reads per cell will be captured in that one kit? And that 1 by 1 centimeter by 1 centimeter chip?
I think this is a good question, but a very difficult question as well. Let me say this way. So in terms of number of million of reads, we do 1.5 billion reads per 1 centimeter by 1 centimeter. And we think that around 50,000 reads per cell is good enough to give you good amount of genes detected per cell. However, the amount of reads is also variable in the sense that sometimes your cells are very packed. In that case, you get much more number of cells, and then you might need to top up the sequencing. If the cells are not that much packed, we don't need to have it. So we recommend on average 1.5 billion reads per chip. You can have multiple tissue on the same chip. So the price, as Craig said, is based on the chip, not on the number of tissue per chip. We have seen in-house that some samples have necrosis happening, then your number of cells are not much, or have a lot of fibrotic aspect into that. So that's also very important to consider here for the number of cells and the number of and the amount of sequencing to be done.
And Pura also has another question. Does stereo-seq capture link RNAs?
Stereo-seq should ideally capture all poly-A ones. So if your link RNA has poly-A, then yes, not all of them have poly-A. So yeah, that has to be kept in mind.
One more question. How thick are the slices?
I think it's 10 micron. I'm not sure. I think it should be 10 micron.
Okay. And this very last question before we hit 10 o'clock. What are the sequencing reads required for stereo-seq? Is the requirement similar to single cell library preparation?
Yeah, I mean, single cell right now, if you look at 10x, they're saying you can do 20,000, but we usually do much more than 20,000. We say that you can do up to 50,000 reads per cell. I think that should be good enough. But I also say that whatever sequencing we have done, we go back and say, look, your number of cells are very high, you might want to top it up. So it's a good thing to keep in mind that your number of cells will determine your number of reads, not the chip 1 by 1 centimeter.
When you say top up sequencing means sequence deeper?
Yeah, you basically, the cDNA is there, you do first pass analysis and you say, oh, we need to do more sequencing. That cDNA is basically sequenced again. And then the data is merged to reanalyze.
Okay. Great. I think we got through all the questions and we're on time. Well, thank you very much, Dr. Kumar. Please leave your video on. So thank you, Dr. Kumar, again for your presentation and for your time. And thank you to the audience for their great questions. And I do apologize that our third speaker, Dr. Sean Huang, is unable to join us this morning due to unforeseen circumstances. But will just briefly touch that Miraxis also provides genomic services through their core in Singapore. And Dr. Kumar, would you be able to briefly tell us about these Miraxis genomic services available for audiences who are interested in with working with the company, with your company?
Sure. Are you guys able to see my slide?
No.
No? Okay, just give me a second then. Okay.
So
is it possible now? Are you able to see now? No?
no.
Oh, I don't know what happened. Sure. Perhaps now.
Now we see your screen. Yeah.
Right.
Yep.
Yeah,
Now we see
okay.
your PowerPoint,
Yeah,
but it's
so,
not in presentation mode. There we
right.
go.
So
And if you could minimize your bar at the bottom. Yeah, this bar, we can, because it blocks your slide. Yep, there we go.
so we have this fantastic discovery multi-omics solution we have where we do epigenomics, you can do whole genome methylation. We use EM-seq here, which is basically using as a NebNextra, TET, and ApoBec as enzyme rather than bisulfite, which is much more tender. It allows you to, and the bias on the GC is much lower than the usual bisulfite sequencing. We also use miRNA. I mean, as Dr. Heng has also told you about our miRNA, the whole suite of profiling we do. On the genome side, we do whole genome sequencing, whole exome sequencing. We have one of the largest sequencer called T10, where we can do 640 samples in 4 days, 30x coverage, whole genome sequencing. That's the capability we have. We can do all kind of transcriptomic like RNA-seq, stereo-seq is a spatial transcriptomic, single cell RNA-seq. On the protein side, we can do feature barcode on the single cell. Soon we will be coming on feature barcode for spatial as well on the stereo-seq. These are some of the things we do. We also do a lot of bioinformatics in-house as well. I think, yeah. So these are some of the available multi-omics service we have. I said we can do spatial transcriptomics. We can do whole genome sequencing, whole exome sequencing, methylation, RNA, miRNA, and we can also do pre-made library sequencing. Let's say you have Illumina adapter in, we can just convert it and do the sequencing on the platform. Yeah, so these are some of those capabilities we have developed over a period of time in-house, and we have been able to serve customers in the US, in Europe, in Australia, and of course in Singapore too. Yeah, and I will stop here. Thank you for your kind attention.
Thank you. So, so for you who are interested, if you want more information about the genomic services offered by Miraxis, please reach out to Craig Morley. And Craig is the Director of Business Development in the USA. His contact will be shown on the screen. Grace, could you pull up that slide, please. Thank you. Right, so these are all our contacts. So Craig Morley, you can see at the bottom, that is his contact. So please contact him if you're interested in Mirex's genomic platform services, or if you're interested in the microRNA screening assays. Yes, and so but if you are a BIDMC researcher, if you are in the audience and you're at BIDMC, you connect with me, because our call has special discounted rates for Mirex's genomic services. And so, this concludes our workshop this morning, actually. So thank you to Dr. Kumar. Thank you to all of you for joining us this morning. And please feel free to reach out to myself, Dr. Kumar, Dr. Slack, who's the director of the core, and Craig again for any questions related to the workshop this morning. And again, we will send a follow-up email. In the follow-up email, you will have all these contacts, and you have a link to today's session. It's being recorded. And so, yes, so thank you very much for joining us this morning. And I will conclude the meeting.